Good morning, students. In today's class, I am going to continue the second topic, biological classification. In today's class, I am going to talk with you first classification of fungi, then we also discuss about the general characters of the first class Phycomycetes. First of all, we will study the classification of fungi. The classification of fungi is based on the characters like morphology of mycelium, and mode of formation, and type of fruiting bodies. So based on these characters, the kingdom of fungi has classified. Let us see the classification of fungi. Classification of fungi. First, we are classifying the fungi into two groups based on the character of sexual reproduction. In one group, the sexual reproduction is present. Sexual reproduction is present, or presence of sexual reproduction. Another group has made which has no sexual reproduction or absence of sexual reproduction. Absence of sexual reproduction means there are fungi which show sexual reproduction, and there are some fungi which are not reproducing sexual. So the fungi which are reproducing sexually are again divided into two groups. The fungi which are reproducing sexually are again dividing into two groups. Here the character is the presence of or type of mycelium or morphology of mycelium. The mycelium is aseptate in one group. Mycelium is aseptate. In another group, mycelium is septate. Mycelium is septate. But here, all the members of this group are having septate mycelium. They have septate mycelium. There is no such a division in this group. In this group, mycelium are septate. The one group or one class coming. Here we study the class Phycomycetes. The members of Phycomycetes have the aseptate mycelium and they also reproduce sexually. Example for Phycomycetes: Mucor, Rhizophus, and Albigo. Another group, mycelium with septa or septate mycelium. Here we find two classes. One is class Ascomycetes and another class Basidiomycetes. The example for Ascomycetes: yeast, penicillium, in the same way, example for Basidiomycetes: agaricus, justilago. And here there is one more group which we have classified or separated from this group based on first, the members of this group are not having sexual reproduction. Means they continue their generation only with the help of asexual reproduction. So that's why this group is said to be the absence of sexual reproduction group. Here the members of this group have the septate mycelium. The class which come under this group is class Deuteromycetes. Deuteromycetes, which we also call the Fungi imperfecti. Fungi imperfecti because of no sexual reproduction. Example for this group: Alternaria, Colatotrichum, and Trichoderma. So this is the classification of kingdom fungi based on the morphology of mycelium, mode of spore formation, and fruiting body. In today's class, we also discuss about the general characters of Phycomycetes. What are the characters of Phycomycetes? We'll discuss now. Class Phycomycetes, which commonly called algal fungi also. Algal fungi is a common name. So let us see general characters of this group. They found in aquatic habitat. The members of this group found in the aquatic habitat, on decaying wood, in damp and moist place. Damp and moist places, or they are obligate parasites on plants. So, first of all, we will discuss what is the meaning of obligate parasites. So, if they are present on decaying wood in damp and moist place, means their mode of nutrition is saprophytic. But here we have see the obligate parasites. So, meaning of obligate parasite is these fungi, they cannot complete their life cycle without the help of living organism. Means they need living organism for their reproduction purpose. And such organisms are called the obligate parasites. So the next character is the mycelium of this group, members of this group. Mycelium is aseptate and cenocytic. The members of this group have the aseptate mycelium and cenocytic. So here I am going to draw the diagram of this aseptate mycelium, which is a tubular structure and it is filled with the many nuclei. Many nuclei are present in the cytoplasm, which looks like a multinucleated structure. And such condition we call the cenocytic. Such condition is called the cenocytic hyphen. Because many nuclei are present in the same cytoplasma and the nuclei are not separated from any septa or crosswalk. Next is asexual reproduction. Asexual in Phycomycetes. Asexual reproduction. Asexual reproduction in class Mycocetes takes place by zoospores which are motile, or aplanospores, which are non-motile. So these zoospores usually found in aquatic fungi. They found in the aquatic fungi. They may have single flagella or biflagella. They may be uniflagellate or biflagellate. And these spores are endogenously produced in sporangium. Spores produced endogenously in sporangium. So then what is the meaning of this endogenous? Let us draw the vegetative structure of this one of the member structure by taking the example of rhizopus. Rhizopus, I am going to explain you what is endogenous. So these are the hypha and they are for the stoloniferous hypha. Stoloniferous hyphae. And these stoloniferous hyphae have certain structures, and these structures are called the rhizoids. Rhizoids help to absorb the food because these rhizoids secrete a substance which converts the insoluble polysaccharides into soluble saccharides. And these sugar or soluble sugars are absorbed by the rhizoids. So they produce a outgrowth. This outgrowth has such a structure. This is called the sporangium. And inside the sporangium, the spores are produced. When spores are produced within the sporangium, this is the sporangium, and these are spores. When spores are produced within the sporangium, such spores are called the endogenous. If the spores are produced outside the sporangium, they are called the exogenous. So this is the structure of sporangium with endogenously produced spores. Next is sexual reproduction. Sexual reproduction. By sexual reproduction, there is a formation of zygospore. Formation of zygospore. And this zygospore is forming by the fusion of two haploid gametangia. Zygospore formation takes place by the fusion of two haploid gametangia. Gametangia. So let us see how zygospore formation takes place. So here also I am taking the example of rhizopus, which is also called the bread mold. Two haploid spores start germinating and produce the hyphae. They produce the hypha. These hypha are cenocytic, means without septa. So one of them is with positive charge and another one is negative charge. So positive charge hypha is usually considered as a male hypha and negative charge hypha is considered as a female hypha. And when they start growing simultaneously, there is a, in between somewhere, there is a formation of some outgrowth. They produce an outgrowth. The same point. These outgrowths are called the progametangia. These outgrowths are called the progametangia, which also have the cytoplasm and cenocytic nuclei. When these two gametangia come very close to each other or they contact each other, there is a formation of septa on both the sides. There is a formation of septa here. When septa is forming, now they are called the gametangia. They are called the gametangia. And the middle wall of these two is dissolves. And because of that, what happens? The cytoplasm of these two gametangia fuse together and it also followed by the karyogame. Means when the cytoplasma fuses, then it is called the plasmogamy. And when these two nuclei also fuse, that process we call as the karyogamy. Plasmogamy is followed by the karyogamy. Because of this, what happens? There is a zygote formation. Zygote formation takes place. Zygote formation takes place. And this one we call the zygote, which has diploid. And they develops a thick wall around it. This zygote may retain with this parental hypha or it may detach from the parental hypha and produce a outgrowth. Zygote, it may detach and produce a outgrowth. When it produces a outgrowth, the structure is called the Zygospore. This structure is called the Zygospore. And this bulged structure is called the Zygosporangia. And within or inside this, zygospores are produced. So these zygospores are diploid and they come out of this sporangium and they have meiotic division and produce the haploid spores. So among this, 50% of spores may be positive charge and 50% of spores may be negative charge. They again start germinating or producing hypha and they may continue or the life cycle with asexual process or they may repeat the same process. So in this manner, zygospore formation takes place in the class Phycomycetes. And this zygospore formation may be two types. It may be isogamous or anisogamous or oogamous. So when two similar or morphological similar gametophyte fuse and zygospore is formed, then it is called the isogamous. Morphologically similar gametophyte fuse and form the zygospore, then that is called the isogamous. When two dissimilar morphologically dissimilar structures fuse together, gametophyte fuse together and form the zygospore, then it is called the anisogamous. So this is the process which takes place in the formation of zygospores. The example for this group already I have mentioned, mucor, and second one is the rhizophus, which we also call the bread mold. And one more example for this is albugo. And this albugo is a parasite on mustard, where it causes the disease called white rust. It causes the disease white rust. So here I am going to conclude this class. So in the next class we continue the discussion of the second class of kingdom fungi. Thank you very much.